one, membranes and transport. Let's just start talking generally about transport. We have two categories of transport that we're going to put all forms of transport underneath. Umbrella 1 is passive transport. Umbrella 2 is active transport. Passive transport is able to use the environmental energy, heat, to do the transportation. It requires the input of no cellular energy. Active transport is going to require the input of cellular energy, and typically because it's going against the natural motion that the system would undergo. So passive transport, active transport. We're going to talk about passive transport first. The first type of passive transport that we're going to talk about is simple diffusion. This is when molecules that are close together in high concentration move to low concentration. Now, the movement of molecules is random, right? Heat is that random motion of particles as they vibrate or bounce around. They're going to vibrate, bounce around, do their thing, but they're going to do so in a predictable pattern because when they are tightly concentrated, they always move to the areas of the system that are lower concentrated. We call that the concentration gradient. So in diffusion, our molecules or particles are going to go from high concentration to low concentration. Now, for simple diffusion, that could be just as simple as my grandma walking in wearing Elizabeth Taylor's white diamonds perfume. That stuff was powerful. My dad used it to scare away raccoons. I could always tell when grandma had come while I was at school, right? Because you could smell her right when she walked in. That perfume was so strong because the perfume molecules were spreading from high concentration near grandma to low concentration anywhere. These same things happen in our cells, right? The oxygen you breathe in goes into your lung cells and ultimately into your blood cells because the oxygen is in higher concentration in the pockets of your lungs, and it moves to low concentration inside the cells and ultimately into your bloodstream. So it's still high concentration to low concentration, and it's just moving through on its own without any help. So that is simple diffusion. The next type of diffusion is facilitated diffusion. This is when it's going to go from high concentration to low concentration, just like in simple diffusion. It's got to be going across a membrane, and it has to be getting a helper to have it go across that membrane. Things like sugars are too large, or other things like salts are too charged to get across a membrane on their own. They need essentially a tunnel to be made through the membrane. Oxygen can just go straight through it. So that's why it's simple diffusion. But sugars are too large, and they need these helper proteins that are literally stuck and floating around inside the membrane. The one that lets glucose into your cells is GLUT4. Right? It opens up, and then the glucose molecule can slip right through that opening. And usually these are pretty specific. Only certain molecules can go through them. Sometimes they have to be unlocked by the presence of another molecule. This is actually one of the things that insulin does, right? Insulin attaches to that GLUT4, which opens it up to allow glucose to go through. That's why people that have diabetes, right? They have blood sugar problems because they don't produce their own insulin, or they don't produce enough, or they don't produce, there's some other issues that come into play. Regardless, the insulin's not doing its job. And because the insulin's not opening up that GLUT4 protein, their blood sugar remains high, right? It's not making it into the cells, or at least not making it at the rate that it needs to. And that creates problems. So that's how I can just tie this to something we've kind of learned about is insulin is that it becomes the key that unlocks that channel protein that lets facilitated diffusion take place. The last type of passive transport that we're going to talk about in this class is osmosis. And osmosis is essentially diffusion of water. And it's water going from high concentration to low concentration. That's what's happening to these potatoes, right? The potato in the pure water, in that case, the water is 100% water out here. It's something less than that. Let's say 90% inside the potato cells. So in that case, the water is going to go from its high concentration outside the cell to low concentration in the cell. That movement from high to low, that's diffusion. But because it's water, we've come up with a different name for it. It's osmosis. And so it goes from high concentration to low concentration. Well, that's going to do is cause those potato cells to have their central vacuole, this little structure here, full of water. And that's going to create a lot of turgor pressure. It's going to press out against the cell wall and make that sturdy and strong. The salt water, on the other hand, let's say it's 80% water, 20% salt. The potato cells are something around 90% still. Well, now the higher concentration is in the potato cells, and the water is going to go from in the potato to out of the potato. And that's going to create this central vacuole getting much smaller. And that's going to release that pressure, and that potato is going to become much more flexible. There's some terms that go along with this. Now, the tricky part is the terms are actually talking about the stuff dissolved in the water. So when we were talking about being in the pure water, that means there's none of these little dots out here. Typically, when we're drawing these things, we depict the stuff that's dissolved in the water. In this case, there's nothing inside the cells. The cell would be 100% water here, and there would be something like 90% water out here, with this dissolved stuff being 10%. Well, that is where the term hyper comes in. There's too much dissolved stuff outside the cell. So if it's 100% water here, 90% because of the dissolved particles, that water is going to go from high concentration in the cell to low concentration outside the cell. This is very much like the salt water for the potato cells, right? It's a hypertonic condition causing water to leave the cells. And that's going to cause that cell to shrink, get smaller. Hypotonic is the opposite, right? Where there's no dissolved stuff outside the cell, and there's plenty inside. So now it's 100% outside the cell water, 90% inside the cell. So that means it'd be 0% dissolved stuff like salt and 10% dissolved stuff. And so what's going to happen? The reason why the salt can't move, right, is it can't get through that membrane on its own. It's not able to diffuse. And so that leaves the water to move through osmosis. And so in this case, there's a higher concentration of water outside the cell than in. And so that means the water is going to move inward. So hypotonic, the water goes in, hypertonic, the water goes out. And that's going to make the cell swell. And then the last one is isotonic. And this is when the ratio is essentially the same. The water is still moving back and forth, but it's not a net difference. And so the water's constant and stable.
Professor, would it be okay if I ask a question?
Yeah, go ahead.
That? Yeah, so is it like always going to be either hypo or hypertonic when you're dealing with like particular what do you call it, solvents, where it's like salt? Is it always going to make it hypertonic or hypotonic, or is it just a certain amount, like there's a threshold?
It's really just if there's a difference. So most of the time with your salts, right, of your body, you're actually probably closer to isotonic. You're probably closer to even, right? It's just when you like sweat, so you lose water, and now water is leaving your cells because it's trying to replace the water that you sweated out, right? Like your body's kind of going to try and reach that isotonic state most of the time. Hypotonic and hypertonic is when it needs to cause some sort of change to happen or respond to a change.
And is the change in the cell membrane, like it's like letting certain things out or letting certain things in? Is that what the change occurs?
For this specific one, it's only the water changing, and it's passing through the membrane. So the water is what's moving, right? So in this case, right, a hypotonic condition, the water is going to move from out here through the membrane into here until it reaches an equilibrium.
Okay, yeah, that makes sense.
So osmosis is just talking about movement of water. Now, if we were talking about diffusion, yeah, we could have this stuff, if it were capable of passing through the membrane. Remember how I mentioned like oxygen can do that? If this were oxygen, the oxygen would move from inside the cell to out of the cell, right? Because oxygen can pass through the membrane. But because it's salt or sugar, it's too large or too charged to pass through. So it can't go through diffusion. And in that case, unless it had a helper, like with facilitated diffusion, we're imagining that's not happening here, then it would just be up to the water to move alone. If both can move, it's probably, you're gonna have diffusion and osmosis happening at the same time. But for these terms that we just talked about here, hypertonic, hypotonic, isotonic, they're only referring to the movement of water via osmosis.
Okay, yeah, thank you. That's clarifying.
Okay, great. I think we talked about last week, we talked about macromolecules. I mentioned phospholipids as one of those. These fatty acid tails are hydrophobic. These charged heads are hydrophilic. What happens is when these phospholipids get placed in water, they naturally form a bilayer like what's depicted here. These hydrophilic heads always want to be facing out towards the water, and the tails want to be, hydrophobic tails want to face away from the water. And so that's how membranes actually naturally form in conditions where lipids and phospholipids in water are present. We also use a similar idea to help take it, like that's why soaps work, right? They have amphipathic natures to them so that they can surround the oily germs of your hands, right, with the fatty acid tails, and then the heads can be all around the outside, and that allows the water to wash away the oily germs and things like that. Okay, these are just talking about what I was referring to with passive transport and osmosis diffusion and facilitated diffusion. Active transport, again, is going to be require the input of cellular energy. So this is typically going to happen when, instead of going from high concentration to low concentration, we want to force the molecules to go from low concentration to high. We want to make them even more concentrated. And that typically is going to require a protein very similar to these channel proteins of passive transport. There's going to be proteins here embedded in the cell, and it's going to be taking that molecule and forcing it to the higher concentrations. Rather than letting it go through high to low, it's going to force it across. And that's going to require the input of cellular energy. And the molecule that is kind of the readily available cellular energy that's, or the cellular energy that's tied to a molecule is in the molecule ATP, adenosine triphosphate. We'll talk about that in one of the next lessons here in just a moment. But ATP is used to force those molecules to go from low to high, to go against their gradient. We also have to use energy in exocytosis and endocytosis in order to bring in molecules from the outside that the cell might want to do. That would be endocytosis. Literally, you have to use the cellular matrix to push the cell membrane out and around and pull that set of molecules into the cell. Or we can go through with exocytosis, where we use the cellular matrix and we're moving it around. That's going to require ATP. We're going to push a vacuole or vesicle, some sort of membrane container. Maybe we have our insulin proteins that were made in the ER. It's going to be pushed out to the cell membrane and then fuse with the cell membrane. In that fusion process, essentially the inside of the vesicle becomes the outside of the cell, joins with the cell membrane, and the inside of the vesicle becomes now outside the cell with that fusion that takes place. And so that would be exocytosis. Again, it's really the movement that's requiring the energy in those of such large things. So. All right, and then here we just end with some key organelles and what type of organisms have those, right? So we've talked about a few of these previously. That bacteria have a cell membrane, bacteria have a cell wall, they have DNA, they do not have a nucleus, they do not have mitochondria, they do have ribosomes, they don't have chloroplasts, they don't They do have cytoplasm. So they don't have membrane-bound organelles. Animals and plants do. So mitochondria, chloroplasts, cytoplasm, nucleus. Now, animals aren't going to have chloroplasts, but they will have mitochondria and a nucleus. Plants will have all three of those. Viruses don't have DNA all the time. Some of them do, some of them don't. Yeah, so that's really different. Plants and bacteria will have cell walls. So some comparing that is useful. So.